So we're talking about cancer. One or two other points. When you go out in the sun is creating changes to your DNA. When you stay out in the sun for a while, your pigment changes. When you stay out in the sun for too long, you get a sunburn. If you stay out in the sun a lot over years and years, you can get skin cancer from continuous DNA damage. Smoking cigarettes increases the risk of lung cancer, bladder cancer, pancreatic cancer, and so forth. So this is just to talk briefly about the Cancer Genome Atlas. The Cancer Genome Atlas was a big project started in 2006 by two groups at the National Institutes of Health in Bethesda, Maryland, to identify mutations that happen in the most common types of cancers. You can learn more about this project by visiting the website listed. The main goal of the Cancer Genome Atlas is categorizing all common genetic mutations in most common types of cancer to be able to understand how cancer develops in the body and to develop better drugs and treatment options for patients to increase survivability. So we're going to soon talk about whole genome sequencing. A genome is a catalog of all the genes needed to make the animal in question. So for humans, it would be a catalog of all the 20,500 genes that we have. Whole genome sequencing is basically isolating DNA from cells and putting the DNA into a single box, a magic box called a sequencer. Here's a picture of a sequencer. We get 3.1 billion letters of DNA from mom and 3.1 billion DNA letters from dad. Remember that we have two copies of each gene. With whole genome sequencing, we read each letter so that we get the DNA sequence of all the genes. We know that about only 1 to 2% of the genome codes for proteins. In the 90s, scientists asked themselves, well, what is the other 98% of the DNA doing? Here we got the term junk DNA. They said, well, the other 98% is just kind of evolutionary baggage from old genes over the last 4 billion years or whatever, from all the creatures that we are descended from. 98% of the DNA is just junk. It doesn't do anything. There are no genes in this 98%. They don't code for any proteins. Well, from the Human Genome Project, we know that this is wrong. We'll talk about the Human Genome Project in a bit. So this is another article, Identification and Analysis of Functional Elements in 1% of the Human Genome by the ENCODE Pilot Project. This is in Nature in June 14th, 2007. There are parts of this ENCODE project which are still going, analyzing other regions of the genome, trying to understand what their functionality are. So over the last 10 years, we've begun to realize that more and more that some of the 98% is really important to tell our genes what to do. Moreover, a considerable portion of this 98% of non-coding DNA is changed into RNA, but the RNA doesn't code for proteins. Now you know that changing DNA and RNA into protein takes a lot of energy. So why are the cells making all this extra RNA if it's not going for proteins? The answer is this RNA is doing a lot of behind-the-scenes work in gene regulation and regulating the coding RNAs and protein modification. So there are a lot of behind-the-scenes work that the non-protein coding RNAs are performing that we were just totally unaware of before the Human Genome Project was finished. And a lot of this will be very important for diseases, common diseases that we see in adults and children as the years go on. And this will be brought out more and more by sequencing more and more genomes. So humans and mice each have about 21,000 genes. In bacteria, there's an old rule that one gene coded for one protein. over the last 20 years, we started to do some genetic sequencing of lots of different animals and organisms. With all these experiments, the scientists were asking themselves, how can we have the same number of genes as a mouse? What we know now is that in humans, one gene does not code for one protein. Obviously, this is the same in other mammals, but in humans, it's a lot bigger part of the picture. In humans, we have a lot of examples how one gene codes for lots of proteins. There's a lot of RNA and protein modification that occur inside the cell. The special RNA that I just mentioned is called non-protein coding RNA. It is the special RNA that doesn't code for proteins, along with special enzymes that allow the coding RNA to code for proteins that do a lot of different functions. This is how humans can have almost the same number of genes as a mouse, but do all the things that we can do, including talking and creating buildings and machines and everything else. So with projects like the ENCODE and other large projects, we're learning that some of the DNA we called junk DNA before is really important in regulating genes and proteins. I talk more about this on my Facebook group, the $1,000 Genome Group. So in this picture on the right, you'll see again that a gene has different parts. And transcription, you get RNA, and then with translation, the parts of the RNA are cut out. With splicing, parts of the gene are cut out to form a mature RNA, and with translation, mature RNA codes for protein. But obviously, in non-protein coding RNAs, the process is stopped before translation occurs. So bioinformatics. I wanted to talk a little bit about bioinformatics. Bioinformatics is essentially categorizing and studying genes, RNA, transcripts, and proteins. Bioinformatics uses different software applications to analyze this information. Bioinformatics studies are done on plant, animal, and human genomes. With computers, we can look at a gene which exists in several different species and see what parts of the gene is the same and what parts are different. We have certain genes that are the same in different animals. Other genes may be different in just one or two small places, but they can have important changes in how the protein functions in our bodies. Two examples of bioinformatics databases include in the U.S. include the National Center for Biotechnology Information at the NIH and the UCSC Genome Browser out in California. This stands for University of California at Santa Cruz. They house very large amounts of data from thousands of research projects over the last 30 years. So these databases are not designed really for medical doctors or patients. Some medical doctors use them, but they are not especially intuitive or easy to navigate in many cases. There's a great disconnect between discovery in the bioinformatics world and translation of these discoveries into helping patients stay healthy. The bioinformatics programs allow researchers to look at one gene for several different species, including humans, to see what changes have occurred over evolution, and to study how these changes work in the body. So we'll stop here and we'll go on to the next video.